Alrighty, good morning. We're going to pick up where we left off yesterday. Um, with your test crosses, these are a different type of monohybrid cross that you can do if you don't know the genotype of an organism. So what happens is you take that unknown organism and you're going to cross it with a homozygous recessive organism. So depending on what the results are, you can then figure out whether or not the parent was homozygous dominant or heterozygous. Obviously, if the parent is homozygous recessive, you know what their genotype is. They have to have both recessive alleles. But you wouldn't necessarily know if an organism that's showing the dominant phenotype, you wouldn't know if it was homozygous dominant or if it was heterozygous just by looking at it. So that's why we do these test crosses. If all of the offspring showed the dominant phenotype, that means that the unknown parent was homozygous dominant. That first set of boxes explains that because the homozygous recessive parent can only pass on the recessive allele, and then the unknown parent would then only pass on the dominant allele because that's all it had to pass on. So all of the kids would show, all of the offspring in the F1 generation would show the dominant phenotype. If you get offspring that show both the dominant and then some show the recessive phenotype, then you know that the unknown parent has to be heterozygous, because the only way for that to happen is for the kids to receive the recessive phenotype kids. The only way for them to show that recessive phenotype is if they got the recessive gene from the unknown parent and that homozygous recessive parent that you crossed it with. And that's what that second box is up there. So that's why you do test crosses. Okay? So we'll try one. You've got long tails on mice are dominant to short tails. When a test cross was performed on a long-tailed mouse, the phenotypic ratio was 4.0. What was the 4 to 0? Sorry, 4 to 0. What was the genotype of the long-tailed parent? Well, since they're all showing that dominant phenotype, the only way that's going to happen is if the unknown parent was long-tailed, or sorry, homozygous dominant. Because remember, since they're performing a test cross, one of the parents is homozygous recessive. The only way for all of those offspring then to be showing that dominant phenotype is if the unknown parent had 2 copies of that dominant allele to pass on. So that's what that third box is, that third cross. All right, here's another one. Green seeds are dominant to yellow seeds on a pea plant. When a test cross was performed on a green-seeded plant, the phenotypic ratio was 2 to 2. What was the genotype of the green-seeded parent plant? So, since you're doing a test cross, you know that you're crossing one parent with the unknown parent with a homozygous recessive plant. So in order for the phenotypic ratio to turn out 2 to 2, meaning you've got half of your offspring being green and half of them being yellow, the only way that you can get yellow offspring is if those offspring get that recessive allele from the unknown parent and the known parent. So that genotype has to be heterozygous, or big G, little g. Because again, if the unknown parent was homozygous dominant, you would have all dominant phenotypes showing up in the kids or in the F1 generation. So those are pretty easy. You don't even really have to do Punnett squares. You just have to know what the phenotypic ratio is, and then you can just automatically tell what the parent was. All right, another situation that we use Punnett squares for is incomplete dominance. This is a this is when an individual will show a phenotype that's kind of a mix between the two parents. The most common form is color of flower and snapdragons. It seems to be the prime example. So what incomplete dominance means is that no trait is dominant over the other. That'll be important in a little while because there'll be one where you've got both traits being dominant. So an incomplete dominance, it means neither allele or neither trait is dominant. They don't share dominance. Like I said, it becomes a blend of the two. So, like I said, it oftentimes we show it in terms of snapdragons. They seem to be the quintessential example. So if you ever hear about incomplete dominance, most of the time it's with snapdragons. And that's because flower color snapdragons, you usually have red snapdragons and you usually have white. There's an intermediate form that shows up occasionally as pink. So in order for you to be red, you would have to be homozygous dominant. Because if you are heterozygous, you get that intermediate or incomplete dominant phenotype of pink. It's kind of a blend between the red and the white. So in this case, in your fourth, I think this is your fourth one, you're going to show, this is your fifth, sorry. You're going to show me kind of, so I'm at a loss for words, sorry. Go ahead and fill out the Punnett square there, because you're going to cross a red snapdragon with a white snapdragon. So a red snapdragon, so the parent on top of the Punnett square, that's going to be homozygous dominant, because if it was heterozygous, it would be pink. Then what you're going to do is you're going to, along the side, if you decide to do the second parent on the side, that's going to be the white snapdragon. So its genotype is going to be homozygous recessive. And then you're going to cross. I really shouldn't ask you for your phenotypic ratio on this one, but you can give me the genotypic ratio and not really the phenotypic ratio. Probably, this is kind of an older one, but probably what I would ask you to do would be to give me what percentage or how many of the offspring would be red, pink, and white, because your normal phenotypic ratio is only going to give you 2 phenotypes, and in this case we have 3. We have red, pink, and white. So hopefully you've crossed it by now, but what you've got now is you should have the red on top and maybe the white on the side, or vice versa. All of your kids should be heterozygous. So your genotypic ratio is going to be a 0 to 4 to 0. And then your phenotypic ratio, we're not really going to do, because that would technically be like a 1 to 2 to 1. Or sorry, a 0 to 4 to 0. So I might ask you what percentage of the offspring are going to be red. You would say 0, because none of them are going to be 0. None of them are going to be red. None of them are going to have both dominant alleles. How many of them are going to be pink? That would be all of them, or 100%. And then how many would be white? That would be 0, because none of them are homozygous recessive. So that's incomplete dominance. Remember, it's no trait is dominant over the other, kind of a blend. So try this one. I'll go ahead and Stop talking for just a moment. You can push pause. Instead of the phenotypic, give me the genotypic ratio, but instead of a phenotypic ratio, tell me what percentage of the kids are going to be having straight hair, how many are going to have wavy hair, and how many are going to have curly hair. So give me what percentages that they're going to have there, and then what the genotypic ratio will be. Okay, so you should hopefully have come up with, you've got a straight-haired male, so that means he's homozygous dominant, and you've got a wavy-haired female, which means she's heterozygous. That's that intermediate form. So your genotypic ratio should be a 2 to 2 to 0. The two kids, if you put the male on top and the female on the side, the two kids on the top, two boxes, those would be homozygous dominant, and then the two kids on the bottom would be heterozygous. So 50% of their offspring should be straight-haired, 50% of their kids should be wavy-haired, and 0% of their kids should be curly-haired. Dad does not have that recessive allele to pass on. So none of the kids should turn out curly, since they would need both copies of that allele. Okay. I think this is the last one that will go. Nope. One more. Okay, so with this one gets a little bit trickier. I probably should have moved, I can move this over a little bit. So this particular problem or set of problems or situation is going to actually incorporate two different versions. The first is multiple alleles, meaning there are some traits that have different alleles. So they're not going to have like the same allele set, so like H's or D's, they're going to have different letters. So if you have 3 or more alleles, you're said to be a multiple allele trait. And so the major place where you're going to see this in humans is in their blood type, because you can have type A, type B, type AB, and type O. Here's where the second version, I guess, of alleles is. It's codominance. So we're going to kind of jump around a little bit, but keep that in the back of your brain, because we'll talk about what codominance is here after we go over blood typing. But type AB is not incomplete dominance, it's called codominance, meaning both alleles are dominant. With incomplete dominance, neither one is dominant. So now here's the thing, here's where it gets tricky. Type A can be homozygous dominant, you can have 2 A alleles, or you could be heterozygous, meaning you've got one A and one O. Okay, so A and B are both dominant, O is recessive. So if you got the A allele from mom and the O allele from dad, that O allele is going to be covered up. So you won't have type AO blood, you're just going to have type A blood. Okay, same goes for B. If you got B from mom and O from dad, you're not going to have blood type BO, you're going to have type B. Okay, but that's also why you can have type AB blood, because those are both dominant, so that's why it's a fourth phenotype. So in order to get, since O is recessive, in order to have type O blood, then you would have to be, you'd have to get both alleles from both parents. Oddly enough, this is your more common blood type, type O blood. So usually the recessive is not common, but in this case it is. So this one gets a little trickier. So okay, with this one, I'm going to set this one up, and then the next one I'm going to have you do on your own. So we're going to cross a man that has homozygous dominant for type A blood. So his blood type is going to be A. If he was heterozygous, he would be AO. So we'll write that at the top. On the side, we're going to have a woman that has type O blood. So in order to have type O blood, she has to have both O alleles. So the possible blood type, so when we cross them, all of their children are going to have AO blood because dad can only give the A. The mom can only give the O allele. So that's why all of the kids are going to be type AO. Or sorry, that's their genotype is AO. Their blood type, then all of them are going to be type A blood. So we can't get O blood because dad doesn't have an O allele to give. We're not going to have B blood because neither one of the parents has the B allele. Same for AB. There's no B allele, so we can't end up with that. So try this one. Set up your Punnett square and try crossing a man that has type AB blood with a female that is heterozygous for type B blood. I'll stop talking and let you work on that. Okay, so your man has type AB blood, so I went ahead and put him on the top there. And then the female is heterozygous for type B blood, so that means she's got a B and then an O. And then when you cross it, the top left corner there, that is their genotype is AB, but that means that they can have type AB blood. The kid on the top right has a genotype of BB, so they're homozygous, dominant for B. So that means that they can also produce kids that have type B blood. The kid in the bottom left will also be AB. No, sorry. The kid in the bottom left, sorry, since she is type BO, so she's heterozygous, so she's B and O. Kid down in the bottom left, sorry, will be his or her genotype would be AO. Blood type would be, and then the kid in the bottom right would have a genotype of B and O. B from dad, O from mom. So they would have type B blood, which we already have. So they can produce. So these two individuals can produce children with 3 different blood types. They can have kids that have AB blood. They can have kids with type B blood and kids with type A blood. But since dad does not have the O allele, none of their children can be type O. So they have a kid with type O blood. We're a little worried because might be looking at the mailman or something. So, okay. The last thing is codominance. So there aren't really any Punnett squares I'm going to have you do with this one. I'm just going to give you some examples, but we kind of refer to this with type AB blood. This occurs when both traits are displayed, meaning both of them are dominant. Or you can have 2 dominant alleles. So it could be 2 different situations. Codominance can mean that you have more than one dominant allele, like in our blood type. A and B are both dominant. And then you can also have it where it just means that both dominant and recessive traits are displayed. So they're sharing dominance at that point. So in either case, both alleles are being displayed in equal numbers. In AB blood, you have equal numbers of A and B. That one's kind of a strange one, but it doesn't really apply to that. But it probably makes more sense in this situation. There's a coat color in horses. It's called roan colored, and so it's kind of a brown color. And what happens is red-coated horses pass on their red-coated gene, and that matches up with the white-colored horsehair. And then instead of the red coat color covering up the white, these particular horses that are heterozygous, meaning they've got one red allele and one white allele, they end up actually producing equal amounts of red and white hairs to give off this brown color. So at first it might seem like incomplete dominance because it's an intermediate It looks like it's an intermediate phenotype, but it appears different because both the dominant and the recessive trait are being shown at the same time in equal numbers. So that's codominance. I will not give you a worksheet today, but you'll have a worksheet on Monday. So you'll be able to go through, use your notes, and then answer some of the genetics problems. So again, if you have questions, please don't hesitate to ask, and have a great weekend.